Jacques Yarmov. I work for BioLabs, and I'm very happy to have you all here tonight in this research center, which is a fabulous place to work in. This first event of a series that we hope to keep running on Thursdays evening, maybe the first Thursday of the month, regularly. BioLabs is an American company. We run innovation places. Incubators would be a shortcut. It's what we would say in French, communauté d'innovation. We have 9 places in the United States. We started in Cambridge, Mass. We have now several facilities in Cambridge and Boston. We have 9 facilities throughout the United States on both shores, on the East Coast as well as on the West Coast. Within our walls, we host about 250 biotech companies. So there are quite a few entrepreneurs that come and see us, and basically we have about 100 new companies every year coming to BioLabs. We are very interested in coming to France, and we have met and we're working with Servier. We have been working for several years. And the first thing we want to do is not say, okay, we are going to put down an incubator and here is how it's going to work. We first want to meet the community and the entrepreneurs. And that's why we have decided together with our partner Servier to start this conference series, the first of which is tonight. Thank you again for being there at this introductory conference.
Health innovation in France. This is a first step. And I'm very happy to announce today that our partnership is going much further than this. So you'll be the first to hear that publicly, although we're going to go speaking loud about this in the coming days. Servier and BioLabs are partnering to open an incubator, and we're going to keep with that language, at least in France. It talks to everybody. On our future research site in Saclay, you may have heard that we are currently building, it's rising from the ground, a new research facility on the Paris-Saclay cluster. There are going to be 800 people working there, mostly, obviously, Servier researchers. And the purpose of that place will be, obviously, to provide new projects for our research portfolio, but also to open to partnerships and collaborations to the existing academic world that we already know pretty well, but also to deep tech companies, to disruptive innovation, to anything that can bring new innovation to patients in the therapeutic areas of interest.
So synthetic biology has been with us since 2004, and the motivation has been multiple, and there is no single definition for synthetic biology, and you'll see a few, and I'll be missing a lot because this is only a brief presentation. Seeing the cost to get to whatever you want, the environmental impact in 2 ways. One because you may use bioresources, and the other one because you may use a bioprocess, or both, and the time to market. One important thing to realize is that the technology is already there, or the technologies are there. The problem is to put them together. We would have, at that stage, reconstituted the way that people have done big objects in the past, like, I don't know, a wheel or something. But, as you are well aware of, the difficulty here is that we need a technical mediation because the objects are nanometric rather than a big wheel. A general remark is that no industrial revolution or evolution can happen without de-skilling, which means in essence that what was possible in only 3 academic labs in the world will become available and possible for thousands of engineers. But we all need more. So now, basically, that's the time for biotechnology, and this is called synthetic biology, sort of a second definition. Another slide on the current drivers. Sorry, it's in French in part. Yes, indeed, some projects will require biology, chemistry, mathematics, computer science, microfluidics, automatics, immersed in the sciences of engineering. But this, again, should not be a repellent. most projects are bidisciplinary. And I think that's where lies the explanation why synthetic biology has been heralded so much in the past years. Because it was, if it were to solve only a little problem somewhere on the earth, we would not be speaking about it here and in other places. So as I see it, biology is one of the most complex sciences that humanity has been dealing with. We are very far in biotechnology from having reached a mature engineering capacity. Engineering 15, 16 years after these beginnings, we have absolutely not reached the degree of standardization that you see in chemistry, in electronics, in computer science, and so on. Now, what's important to realize is all of this is work done in academic labs and sometimes also in the private sector, are contributing to the central thing here on the slide, to a set of concepts and methods. And this set is really the heart of what synthetic biology is and explains why it has been heralded so much. Now, from these methods, from these concepts, you can address different. Remind you, if you have not heard that synthetic biology has been active in allowing to develop some products and applications. 5 years ago, the Woodrow Wilson Center in the United States have compiled I think 116 applications or products that were close to the market or on the market. And this tells you that it's now, 5 years later, quite difficult to produce such a list, which is a sign that synthetic biology is moving on.
Thanks to that, from the maybe 1990s, where this human genome project was initiated to sequence all the human genome. We took 3 or 2 decades and $3 billion to sequence only one genome. Now, today, 2015, 2020, it costs only a few $100s, and you can sequence many genomes in just one day. So, so this unlocks also many different applications. So this is about reading the genomes. So once you know how to read, what you're going to try to do is to write it, to enable basically making accessible the design, synthesis, assembling, and testing of genomes, whole genomes. And the goal is to, by the next 10 years, to have a 10-fold decrease reduction in cost and in speed of doing so. And this also includes large-scale genome editing, which I'm going to talk about now. So if we can unlock this powerful... So eGenesis is a company that's spun out of the Church Lab as well. I've been lucky to work with these 2 amazing people, Luhan Yang and Mark Well, that were my mentors. Maybe you've... now, there's obviously hurdles, they're easy to do. So there's one big hurdle is immunology. You need to make this be compatible with human. So how do you do that? There are many new inserts, knock-ins to do, knockouts to do. For instance, genes involved in rejection, immune cell immunity, such as TCRs, histocompatibility factors, coagulation factors, complement, and so on and so forth. I need to do like many tens of edits to be able to make that happen. And so this is what eGenesis is doing using the Cas9. So probably familiar with it, a nuclease that has the DNA, and is guided by an RNA by hybridizing to the complementary sequence in the genome. So they succeeded for the biology part, and they're working on the immunology part. And now eGenesis has raised more than $140 million in the US, recently $100 million. And instead of cutting the DNA into pieces, it basically changes only one letter, either C into T or A into G. But when we modify those editors, we could do thousands of edits in a single cell. So we reached a point where it's feasible now to get to that point. And these companies from 2018 have a lot of application in different fields of synthetic biology. So protein engineering, cell therapy, DNA storage, and so on and so forth. So there's definitely a big push towards synthetic biology that will explode. I mean, already exploding, but will be very helpful to have more people working on all those guys.
And so the technology needs to be applied to unmet medical needs. So that's how you can actually engage with big pharmaceutical company. So that's a key aspect. And to do that, you actually need to make sure you understand what the company you're addressing, what their interests are in terms of therapeutic areas or disease that they're pursuing commercially and in development, because it's hard to enter a new disease area as a big pharma. You have to invest a lot of capital and human resources to...
Not oppose anymore big pharma versus the rest. I think it's probably, the world today has changed, and if you look at the pipeline from different companies, and even the biggest companies, where the biggest drugs are coming from, right? I think they are coming from outside. I think the small companies are now, and external innovation is clearly, I think, a big driver of wealth for the big pharma. So, and there is also a lot of flux of changes in people. A lot of people, scientists, moving from small companies to big pharma, and something from, and you have seen also the reverse. A lot of people in big pharma being also wanted to escape bureaucracy and going to small companies. So this dialogue now is changing, and at the end of the day, as we said before, in the interest of the patients, as we say, good medicine will be good business. I think foundation of what we are doing in science. And what do you start with? The question, right? What the synthetic biology questions, or at least want to address in terms of key questions. Where we will be today, I think, without having the question about, I think, immune system and cancer, without now having the CAR T cells, I think, discovery, for example. I think that would be fun, I think, and the question was clear. So science is driving, I think, absolutely this dialogue first. So the quality of the scientific dialogue, I think, is the first foundation. There is no business in pharma industry.
As a VCs, most of the time companies are failing because of people, not science, because of shareholders are not agreed altogether, because VCs are not agreed altogether, etc. So, of course, science is a key point, but at the end of the day, when we invest in a team, and sometimes science is not as good as we planned, but if the team is good, management will evolve, will switch, will repositioning the technology, and perhaps we can save all the company. And I often say, all the investors are not for all the entrepreneurs. Sometimes you can work with someone and don't work with another, so that's it. And of course, it's a matter of trust.
All the off-target effects that are occurring. If someone is coming to tell me, okay, tomorrow I have this key technology that ensures that my guide RNA will be extremely specific with 0 off-targets, then I say that's a huge benefit, because you do improve significantly the tolerability of this new modality. Or if you have a way to overexpress the Cas9 just like a tiny bit amount of time to do the job one time and not multiple times. And then that type of technology can translate quite easily into benefits and into something that will significantly differentiate yourself to the current technologies.
When we look at the technology, we usually, such as a synthetic biology application, we usually look at how validated the technology is when we are ready to license it. So if we have in our own pipeline a clear idea of how we want to apply this technology to our own therapeutic area, we're not so interested in what type of indication the company has pursued to advance their platform, but more how far they advance their platform. So,
and this is something that we have to certainly not only to praise, but I think to continue to fuel very much, because we are in a world of ideas, and whoever has an idea is always a good idea. I think until proved otherwise. Again, we don't know what we don't know, but your neighbor may know. I think in the next lab, I think and that's why this is the beauty of your concept, I assume.
The startup is great, gives you the whole environment, and you will be really focused on it. So you might think that it might somehow be easier as compared to big pharma, as you said, that have lots of process that are not used to understand this type of new technology. But in the meantime, when I did my interview in Servier, and it's true, when I joined the company, you had everything. You had from the excellent expert in protein biology, in screening technologies, in animal modeling. That's for the science part, but you have also all the in-licensing technologies, the patents. And I realized that because my intention was to be able to bring this dream true, where would I have the most chance to make it happen was really in the best environment where you had most of the things that could help you, but also, as you said, the human side, where the people are here also to believe in you, to follow you, and to support you to be even better and achieve this dream.
Something comes to mind immediately is risk. We are in a very risky business, guys. I think 90+% of the project will go down. This is certainly a message I have for you guys. It's about don't avoid risk. Don't avoid risk, but manage it.
But I think at the end you have to listen your red flags, because you are like married people for 5, 6, sometimes 10 years, and we will have up and we will have down, and if you're not aligned, it will be a nightmare, and it's experience, in fact.
It also works when you actually talk to a future partner. So whether it's both ways, meaning if you are going to start collaborating or even like developing a product together and there's no chemistry between the development teams or the research teams, and you feel there's, they're not speaking the same language, they don't want to spend time discussing science, you know that probably the partnership will maybe happen, but then the science will not progress the right way because the R&D teams will not be discussing and productively collaborating. So, yeah.
But think about, always like drug development, keep the end in mind. What's I think factor, what this product has to do, I think will do in the future. And all your development strategies and execution will be just plain reverse engineering. That's it.
Because we are not experts on your technology. You are the expert. And you have the right also to guide us to understand better your technology. Because each time you review a project, we do look deeply into it.
Horizons in the conversation is already one thing that we can do. In France, we have, when I left France for the US, there was like probably 5 biotechs around, or maybe 10. When I came back, there were much more. And just like building ourselves, the ecosystem is the only way that it's going to happen. So we all have, as part of the ecosystem, a role to play to foster it, to make sure people are attracted to these, roles, whether entrepreneur, whether VCs, whether any type of role that needs to create a fruitful and productive ecosystem.
Clearly, we are at a juncture where we are starting to see the effects of the policies in public research in France now. That is the negative effects. So the public and academic research in France is going down and is visible now, even though we clearly see that it started in 2005, 2007, this process, but now it's visible. We are facing this difficulty, it's clear. But this also is improving a little. It's just improving perhaps a bit too slow. And when I'm talking about the United Kingdom, I avoided to talk about the United States, which is still 10 times above the level in the United Kingdom of available investment funds in biotech. So I think this is really part of the deep reasons. But I wouldn't say that the situation is not improving. It is improving, and it will improve thanks to BioLabs. Thank you.
Well, thank you.
Question: how do you bring this type of change and bring entrepreneur and more innovation? I realized it a little bit later, is just do it. Do it like the Americans are doing, quick and dirty. You fail, but you learn with your failure. You learn on your dirt, say, to improve it, to make it a little bit better. And instead of planning for the perfection, which at the end will probably not be perfection, you just do and learn from it.
Be the change you want to see. That means you are also part of it. That means don't try to find somewhere in the French administration and government and politicians. By the way, don't expect anything for politicians, never. But I think you initiate the dialogue with in your department, with the other scientists, with the heads of departments. Guys, if you don't change the world, who will change the world for you?
Questions directly. I would like to invite everybody to first thank our panel and then meet them with a glass in hand. Next conference, don't hesitate to come.